Hello everyone. In this video, we're going to review the cytoskeleton. The cytoskeleton is highly complex. And so I'm going to try to just focus on the main things, especially the things that I would want you to know for an introduction class about the cytoskeleton. There's a lot of information about muscle movement, which eh, it's not necessary. I mean, it's important to know, but I'm not going to focus that much on it for at least for this chapter. You're not going to have hardly no questions from your exam about that. So in this image here, this is actually a cell. And so the big blue blob here in the middle is the nucleus. And the red and the green represent the cytoskeleton. So what is the role of the cytoskeleton? It's support and motility. The cytoskeleton, I like to think if you're building a house, if a cell was a house and you're building a house, the cytoskeleton would be the walls, the wood that forms the cell. The cytoskeleton helps to support the cell and maintain its shape. It interacts with motor proteins to produce cell motility. It allows for cells to be able to move. Inside the cell, vesicles and other organelles will use motor proteins that are what we call feet to travel along the tracks provided by the cytoskeleton. One of the things that I remember I never could quite grasp when I was learning about cell biology was that I thought organelles just floated through the cell, but no, that's not true. They travel along um, roadways that are created by um, the cytoskeleton inside of the cell. So it's very fascinating to know and learn. And so these roadways here is an example of a microtubule roadway, and you can have vesicles that will travel along those roadways using a motor protein or the feet that will pull them along the roadway. So in this image here, you have a microtubule road, and then you'll have vesicles that will attach to it and travel along that roadway. Now, there are three different types of fibers that make up the cytoskeleton. There are microtubules, which are the thickest of the three components of the cytoskeleton. They're big, huge, hollow tubes. Then there are microfilaments, which are what we call actin filaments, and they're the thinnest components of the cytoskeleton. Then you have intermediate filaments, which are fibers with diameters in the middle range. They vary in thickness. They can vary in thickness, but they're not huge hollow tubes like the microtubules or just single strands like the microfilaments. And so in this table here, it gives a really nice, good summary of the three different types of components that form the cytoskeleton and their function, their role in the within the cell. So let's go over them. There's the microtubules. There are huge hollow rods that are about 25 nanometers in diameter, and they could be up to about 200 nanometers to 25 microns long. So uh, that those numbers don't really mean anything if you don't understand how a nanometer is, but they can span for a portion of the cell, or they can span quite huge long distances like across the whole entire cell. Microtubules are constructed of these dimers of tubulin, and tubulin is what forms the proteins that form the tubes. Functions of microtubules: they help maintain the shape of the cell. They are the guiding movement of organelles. We mentioned a while ago the roadways. So microtubules will literally form the roadways of the organelles. And one of the important things that microtubules do are separate chromosomes during cell division. That separating of chromosomes during cell division is a very important role. And in animal cells, Microtubules will grow out of and form a centrosome near the nucleus. The centrosome is this very large microtubule structure that will form, and in my mind, I think of it as kind of like a crank. Microtubules will attach to the chromosomes, and during cellular division, it will pull those chromosomes apart. And the centrosome will, to me, is like this crank system where it pulls the microtubules and separates the DNA in the cells. We're going to cover that in depth when we get into chapters 12. In animal cells, the centrosome has a pair of what we call centrioles, which has nine triplets of microtubules arranged in a ring. Other eukaryotic cells can organize microtubules in the absence of centrosomes, but they usually do that with centrioles. Plant cells do not use centrosomes and centrioles. They are a little, plant cells do things a little bit different, and we'll talk about them more when we talk about cell division. So this here is showing you the centrosome, which are the centrioles here, and they are will be used to form for cell division. You can barely see the nine ring, double ring structure, which I will cover that in um, the next slide when we're talking about the structure of cilia and flagella. They still have the same very similar microtubule structure. Microtubules are also important in cell motility. Microtubules control the beating of flagella and cilia. Microtubules containing extensions that project from some cells form the cilia and flagella. Many unicellular protists are propelled through water by cilia or flagella. And if we're talking about human cells, the human sperm cell has a very large flagella uh, for motility. Cilia are found in a large number of cells on the surfaces. A lot of the linings within your body, your intestines, for example, are covered with cilia to help move things along in a certain direction. Cilia and flagella differ in their beating patterns, which that's not too important, but both of them are still responsible for um, motion. So here you can see flagella, which are responsible for the direction of the swimming of the sperm. And then you have cilia, which are responsible for organisms' movement. You can have a power stroke, and then the recovery of that stroke takes place. Now, cilia and flagella share a very common structure. The group of microtubules sheathed in an extension of the plasma membrane. There's nine doublets of microtubules that are arranged in a ring that surround two single microtubules in a center. This nine doublet and two single microtubule structure is attached to what we call a basal body that anchors the cilium and flagellum to the cell. There are motor proteins called dynin, which drives the bending movements of a cilium or flagellum, which causes them to move and beat. So this here is showing you a cross section of a cilium. And so if we was to do a cross section of that, you would see the 9+2 structure. So you have nine doubles and then two in the center of the central microtubules. Now, there are proteins called dinins that are attached in between these and to the central microtubules. Now, the dinins form these links between the microtubules, and it is the contraction of those motor proteins that allow for the whip-like motion and movement of cilia and flagella. Now, the next group of filaments we're going to talk about are the microfilaments, or commonly called the actin filaments. Microfilaments are solid rods that are about 7 nanometers in diameter, built as a twisted double chain of actin subunits. So there is a network of microfilaments throughout the cell, and they help support the cell's shape. They form a cortex just inside the plasma membrane to help support the cell shape. Bundles of microfilaments make up the core of the microvilli of your intestinal cells. So this is showing you have your plasma membrane, and then right outside of the plasma membrane, there will be layers of actin filament, and then there's intermediate filaments also as well. Microfilaments are also function in cellular motility, contain the protein myosin in addition to actin. So cells will crawl along a surface by extending what we call a pseudopodia, which is like a fake foot, and moving towards them. Cytoplasmic streaming can be found in plant cells, which is this is a circular flow of the cytoplasm with the cells, which is driven by an actin protein interaction. Now, the whole purpose of this is just to show that because of the cytoskeleton, this network of filaments, cells are able to do and be motile. It's not static, things are not held in place. You can have, for example, your muscles, the muscle cells are moved based on the actin and myosin motors, proteins. Um, which I'm not going to go in a super in depth about that. Cytoplasmic streaming, which is it's the flow of the organelles within plant cells and other cells that allow for movement. To think of it, if you're a chloroplast, you are constantly in need of being moved, or you might be need to be moved towards and orient more towards the sun. So that's important. And and then you have organisms that are constantly moving and changing shape. For example, amoeboid movement in an amoeba, where they're able to extend what we call a pseudopodium, like a fake foot out, so that it can search and explore. The last type of component that forms the cytoskeleton are the intermediate filaments, and those that range from 8 to 12 nanometers. They're larger than microfilaments, but smaller than microtubules. Um, Intermediate filaments, we think, are more permanent structures inside of the cell. They support the cell shape and they help hold organelles in place, but they're not as readily actin filaments can be assembled almost instantaneously, and microtubules are that can be built and broken down whenever they're needed. And so intermediate filaments just have a more static role compared to the other two. And that covers the three different components of the cytoskeleton. In the next video, the last video, we're going to be looking at some extracellular components and things that form cell communications between cells.